Computer. Here we go. Okay, good. All right, thank you very much. Now, okay, family. Okay, there goes John. So now we got everybody, and it's not but 3 or 4 minutes after, so that's not too bad. Okay, we got 8 participants. That's including me. So we got everybody. Now let me go back and re-share the screen. I thought it would be good. This is not the starting slide for the 8th session, and but it is something that we talked about last time, and I'd hope to finish plant physiology today. I don't think we'll quite do that, but the last 4 or 5 slides we might not get to, but we'll see how it goes. But this slide was back when we talked about totipotency and getting the right ratio of auxins to cytokinins, which actually turns out to be almost a constant. And this ratio here is the one that promoted stem and root growth when you got the right combination. So now, having said that, we're going to go to the starting slide. Let me see if I can minimize this a little bit. You know what I need to get. Yeah. All right. Now, it's got me talking and not me. Well, actually, in the past, I've had myself in there, but I really don't need myself in there. So I got me talking, and that's good. Let's go on down. I don't know why I'm having so much trouble navigating. It's not usually. Maybe I better go to a tutorial for Zoom. Anyway, here we go. This is where we're going to start. This last item we may not get to, although we'll talk about it, and as the last few slides in this segment. My favorite topic is the next one is plant reproduction. It's not because of the sex, it's because of the fact that it's not memorization like people think it is. I guess that's why I really like it. Let's see if I can fix this. I kind of like this. All right, small show active speaker video. Okay, there's Donna. Okay, I don't know why it's showing you, Donna, but let's see if I can. What's that show? Thumbnail video. Okay, no, I really don't want to show this for the recording. Show grid video. That's even worse. All right, now. All right, let's just be content with me talking. Okay, all right, we got 8 participants, and when I'm sharing this script, I'm going to go up and fix this again. Sorry about all this. I just feel retarded. I get this for some reason, I feel like I'm dragging an anchor today, and I guess most of it's around my stomach, but I've lost about 15 pounds, but you never know it. But anyhow, all right, plant growth and development. We've talked about the first 2 topics. Now we're going to talk about number 3 and number 4, and probably not get to number 5 today. Let me make it on the enlarged. Okay, now, see if I can get this little bar out of the way too. All right, now let me get to I guess forget it. We'll probably go next week. There's nothing that stands in the way, I hope. But now the principal plant hormones / growth regulators. We're going to find out in the next slide at the bottom left, which is a concept slide, that there's 3 primary sets of control factors, and it has to do with chemical substances, that's the hormones and the regulators, environmental factors, and the genes. As in humans and other animals, and in certain genes are active in certain cells at certain times in the life cycle and not other times. Some genes apparently never get turned on. But the major factor that regulates growth of any kind is genetic information in the nucleus of a cell. And let's see. Okay, this is a concept slide, and what I was just trying to say was that bottom left, those answer the question, what controls growth and development? The genetic information, the hormones that the genes are responsible for producing, and the environmental factors that take part, and these could be microenvironmental factors too. But the bottom left is what controls growth and development. Now, the concept diagram here has three major themes. That's when it comes to plant growth and development. You've got control, you've got the plant responses, and then you got the timing of growth and development, which is the topic we probably will have to defer to some extent. We'll see how it goes. As for control, we'll address the three factors with hormones first, then we'll talk about the responses to the hormones. But now we're going to deal with a few more concepts on this concept diagram. Genetic control at the left is intra, I-N-T-R-A, intracellular. That means it's within the cell. And hormonal growth is usually the growth itself is usually intercellular, but unlike animals, plants don't have a specialized hormone producing set of glands. They don't have adrenal glands, thyroid glands. In other words, there's no such thing as endocrinology when it comes to plants. That's a topic for the animal world. But plant cells can typically or often respond to their own hormonal productions. And sometimes the cells adjacent to them. There is some like transferring of auxins from the leaf to the apical meristem and something like that, but that's pretty short. That's not long range transfer like what happens in animals. So there is some transfer of hormones from the cells that produce them, like in the leaves, to this apical meristem of the stem or root. But there is no set redefined glandular tissue system that produces hormones, okay, unlike animals. Now, why do plants generally respond to stimuli? This must be in the question that's everybody's mind one time or another. Well, everybody in this class, I don't think the public gives a rip, but why do plants generally respond to stimuli more slowly than animals? And the answer has to do with the fact that plants do not have a nervous system or a muscular system. And I've already said they don't have a dedicated hormone producing set of tissue. The cells can do it without being in the form of a gland. Now, on this slide is in the center in terms of responses are tropisms, which is Greek for trophic, is Greek for change. And it refers to growth. And nastic movements are not growth movements, but they're talked about when we talk about growth movements, because that's the only appropriate place, because nastic movements have one thing in common with tropisms. They are a response, okay? They're a response that plants have. They're not growth movements. In fact, nastic, trying to remember what the Greek is, and I wrote it down somewhere. Okay, nastic is a word that means closed, and you'll see an application of that in a minute. In Greek, it means closed or solid, but closed, the context of closed, we'll see as we go along. Well, let me go ahead and point out three primary differences between nastic movements and trophic movements. Unlike tropisms, nastic movements are independent of the stimulus. It doesn't matter which direction the stimulus comes from. That's one difference. Second difference is nastic movements typically are quite quick, happen quickly, unlike tropisms. We already said why tropisms are slow. There's no muscular nervous system to support it. But nastic movements, we'll point out later, are turgor movements, water movements, often happening within lagoons that have a pulvinus, but not necessarily restricted to lagoons. Pulvinus is a swollen portion of the petiole. Those who were on the field trip this weekend saw an example of that, or this past weekend. Some of you were. Now, and speaking of that, everybody's seen the field trip new policy where we kind of let the leader decide, and 10 is no longer the restricted number. The third and final difference that I'll point out between nastic movements and tropisms is that in a nastic movement, they will return to the former condition or state. In other words, if the leaves were open, when they drop down, they'll come back to that. They may not come back as quickly, but they'll return. In tropisms, they don't return to their former distinction. They're fixed. All right, let's see. This one here, one of the maybe not so obvious part of the question has to do with when would plants flower, and that has a lot to do with what's going on down here in the bottom right. Photoperiodism has to do with some plants will flower when they're independent of the light. Some plants will come into flowering if there's a certain number of, it's a short day in terms of light hours, and then others will flower for a long day over a certain length, for example, over 12 hours. And phytochrome is a pigment that gets involved in that process. Phyto means plant, chrome means color. And that'll be mostly a topic for the first 10 or 15 minutes of next week if we don't get to a day. But I'll go ahead and say now that some vernalization, vernal means spring, but the term means cold treatment. Some plants respond to, lilac is famous for flowering. Doesn't do well in the South because it doesn't get cold enough. Some plants flower in response to both photoperiodic requirements, light requirements, and vernalization. So it's like an experiment with more than one variable, which makes it interesting. Any questions? Take that as a no. Now, okay, let's see. The timing question will be addressed later, but we'll take a closer look at hormones and responses of plants to them later. Let's see what else. Okay, all right. This timing business and seasonal features are related to environmental cues. That's what the connection here is. I had lost my place for a moment, but yeah, the connection between that dotted line. I often say that any organism is what it is for two reasons: plants, animals, anything. You're what you are for the genetic information and the environmental effects that the environment has on the genes. Two identical twins can be different depending on the environments they're exposed to. And so now when you add the hormone aspect, it's just like an intermediate. Genes direct that hormones be made, and the hormones direct that things be done. And this happens differently in what environment. It could be an aerial stem environment or a root environment, because that's like a microenvironment. When their stem is in a different environment than the root, and it's kind of, you say, well, they're in the same place, but that's habitat. So there's microenvironmental differences that can be incorporated into this manifestation of traits. And let's face it, a gene is that part of a DNA molecule necessary to make a particular protein. And the protein usually involved is an enzyme. Enzymes lower the energy of activation of reactions and make reactions take place more effectively. That's what, so those are the connections there between the bottom three. Now, well, that'll be more next time. And the next slide, we're going to talk about the hormones themselves. Now, I'll give you a chance to read that. I'll have to take a sip of coffee here. All right. Now, transported to another part using controlling growth and developmental functions. Now, this transport business is something we see in animals, but the transport distance here is not very far. For example, from the hormone can move from one place to another. Could be in the leaves and move to the stem. Here it's has to do with flowering going on. And we've already said that there is no organ, no system, no hormone system, unlike animals. There are receptors, and receptors are proteins that bind very specifically to a certain signaling molecule, like a hormone. And ultimately, signals like hormones cause changes in behavior because they cause changes in gene regulation. I'll repeat that. Ultimately, in this process, signals like hormone signals cause changes in behavior, and the behaviors can be slow in plants because they cause changes in gene regulation. Now, this is an important concept, and I'm going to let you write it down if you want to. Gene regulation is the process that causes cells to access certain genes and not others. Okay, that's what gene regulation is all about. It's a process that causes cells to access certain genes and not others. Okay, so sending signals that change cell behavior. That's what this slide's all about. Now, this is all right. A phytohormone concept map, and it's not immediately obvious from the concept. If it's not immediately obvious from this concept map, I'm going to point out certain things. And one of them, the first one, is that more than one hormone may influence a single plant response. And we saw the opening slide today that I showed you from the callus development is a function of that, where you had you had the auxins and cytokinin ratio, auxins and cytokinin ratio affecting the development of the callus. And the location of where in the callus it was had a lot to do with it. In the apical portion of the part where the light was developed in the stem, the part down in the substrate developed into a root. So it's that's a factor. Now, this kind of reminds me of the way hormones work. It reminds me of the way genes work. In humans, or actually in animals, or in organisms in general, one gene can control one reaction, but one gene can often control several things or reactions. So one hormone can affect more than one trait, and this we're seeing examples of that. Now, a second factor is two or more hormones interact in many responses, and I actually already alluded to that. Two or more hormones interact in most responses. That's like the auxin cytokinin thing. And last, what a hormone does depends in part on the target tissue, and that's exactly what I meant about the callus. The basal part of the callus will develop into a root, and the apical part of the mass of cells, or callus, will develop into the stem. So that is, hormone may have different effects on different tissues depending on the location of those tissues. And the location of those tissues is a manifestation of microenvironment when you think about it. Now, lastly, Florigen has a question mark because we still, as far as I know, no one has ever identified a Florigen, a chemical that actually promoted flowering. They know about 3 genes involved, but that's all they know. And I haven't been into plant physiology in any depth in recent times, so maybe there's something coming down, but I think I would have heard about it because I do get this newsletter about that talks about new developments. Now, we'll go ahead and talk about this much about Florigen and get it out of the way. We don't, again, botanists, and this is coming from page 176, Botany for Dummies, that book, one of the books I talked about. Botanists still don't understand all the mechanisms that induce flowering, despite identifying 3 genes that are responsible for it. The exact Florigen compound is still a mystery, if indeed it is one compound. We do know about phytochrome, a pigment that's it has to do with flowering, but in long day and short day plants, but that's just those plants. Now, when we focus more on timing and growth, which will probably be next time, we'll talk more about phytochrome. So florogen, or also known as flowering compound, is the term used to describe the hypothesized hormone-like molecule responsible for controlling or triggering the flowering in plants, and that means non-long day and non-short day plants, which is, we do know phytochrome's involved in that. Florogen's probably involved in that too. We do know, and this slide tells you that the florogen is produced in the leaves and acts in the apical meristem and changes that meristem from a vegetative growth to a reproductive growth. Now, there are plants like buckeye that has a bud that's mixed. It has vegetative and floral parts in the same bud. That's the exception. Let's see. Second bullet. Okay. The second bullet. I don't know how much anybody, well, John might down in Florida, but grafting, florogen is graft transmissible, even functions between species. But the best example of it that I can think about when it comes to this is the whole citrus industry in Florida and I guess California depends on grafting, where you take a very hardy rootstock that's not susceptible to nematodes or other plant pathogens, and you graft on it a different strain or different slip, I guess they call them, that they stick into the basal part and they grow oranges. So an orange is kind of like I can't think of the word, but anyway, it's a combination of different strains, and the one that tastes a certain way is grafted into the part that grows without getting disease. If you take try to grow regular orange totally from in the ground, it would be subject to all kind of it would not do well, and they figured this out. There's people that make big bucks that run around and do this, do a whole field manually, go around and insert one strain into another in the which is really grafting on a big scale. All right, I already said the last one, so there's no need to repeat that. Besides florogen, though, there are better known compounds. Now, if you look 1, 2, 3, 4, 5 on the left, these are the same as what we have here on this previous slide, with the exception of florogen. So the first 5 are the same as these 5. This is a table that goes about looking at the different kinds of hormones. There's 5 major, ignoring florogen, there's 5. This table summarizes 5 major hormone types, and then auxins is the first. Now, indole acetic acid is the best known of the auxins, and auxins in general, they influence many actions, including bud formation, sex determination, and fruit development. You see, that's quite a long list over there on the right, and they operate by causing cell elongations. Cytokines down here that work stimulate cell division. And I've underlined those because those are the 2 that are involved in that opening slide with the callus development. So you can see why auxins and cytokines, the right ratio, which is a constant, they tell me. The A/C, the auxin/cytokinin ratio, that opening slide showed it is a very precise number that physiologists have determined, the ratio of cell auxins to cytokinins, A to C. Auxins also promote the production of other hormones. Sounds like the thyroid gland, doesn't it? And they control the apical dominance. I just cut in fact, I had a tree limb that some of you saw. It destroyed the apical meristem of my swamp white oak. And now that I took that apical meristem out, the lateral branches are starting to grow and changing the appearance of the plant. We've already said that these kind of auxins can convert stems from vegetative apices, apical meristems, to reproductive apical meristems. And the way that first underlining there, auxins stimulate cell elongation is they increase cell wall plasticity. Cell walls are normally rigid, but the auxins make them more flexible. The auxins also stimulate cambial, that's meristematic cells. We know about what that is, like in the cambium, to divide, and in stems cause secondary xylem to form or differentiate. Auxins can also promote lateral roots and adventitious roots. Adventitious roots are the roots that develop from some other place than the primary root. Like most of you have seen poison ivy growing up the side of a tree. Those are adventitious roots because they come off the stem and not the primary root. So an adventitious structure, and we talked about this, but as we talked about a lot of stuff over this course, and some of it's months ago now, but an adventitious organ is something that develops from an origin that it would not normally develop from. Also in this, I don't know if I got it in the upper right-hand corner or not, but auxins in seeds regulate protein synthesis, and they develop within the flower after the flower is pollinated, causing the flower to develop fruit that contains seeds. And let's see, what else about auxins? And indole acid is, I'll show you in a minute, it's very similar to tryptophan, a compound called tryptophan. Now, let's see, on this slide, the cytokinins have already told you function in cell division. Most of you, especially John, being in plant pathology background, know that galls, G-A-L-L-S, are plant structures that the insect is caused to manufacture for it. So a plant gall is a growth response to an insect parasite, but cytokinins play a big part in that. They actually delay senescence and provide the insect with a winter home. So that's pretty crafty interaction between the insect and the plant. It's not doing the plant much of a favor, though. We'll see all right, gibberellic acid, which are first discovered in fungi, we'll talk about in a minute. They do cell elongation too, like auxins, but they also do something that's not shown here. They help in fruit development. And you'll see a slide of that showing how grapes can respond to gibberellic acids. One last thing about cytokinins is florists use cytokinins to keep their cut flowers fresh. Okay, and lastly, we'll talk least about these other two, abscisic acid, and ethylene gas, but I will go ahead and say that the abscisic acid, before I forget it, is what's responsible for the leaves from abscising from the stem. That actually is a function of in timing, like photoperiod that we talked about, where environmental effects get involved. Plants don't normally in the temperate zone drop their leaves due to cold. They drop their leaves due to the decline in the photoperiod. Deciduous trees. Tropics is different, but a lot of temperate trees. The result is that a tree next to a street light with artificial light may keep its leaves longer, typically will keep its leaves longer than those leaves on the same tree away from the street light. So what I'm describing is the formation of an abscission layer that may be retarded due to the extended light period. So the environment gets environmental factors get involved in with the hormone. And ethylene, some of you are already familiar. What happens that is when you put bananas can be a good example. We'll show you a slide of that and how fruit development takes place and how they get apples to ripen by spraying a whole warehouse of them with ethylene gas, and they may look good, but they don't taste too good. And that's the sorry part of that story. Now this is I guess everybody's still there. This time I can't really. I think I'm going to go to the tutorial. Okay, here Donna in the background. That's good. All right, now.
I have a question, Bobby.
What's that, Ronna?
I have a question. The
Yes.
abscisic acid, is that something?
Wait a minute now. You're going to have some reason I'm not hearing you. Go ahead. I'm hearing you, but we weren't very loud.
Abscisic acid, is that something that they spray on plants to keep?
What's that? Which one?
The abscisic acid?
Abscisic acid? No, it's not. That's ethylene gas.
Well, is that the acid though that they use? They spray it on plants to hold them at a certain development, like flower development for.
No, that's not abscisic acid. When you abscise, you cut off, and that's what the word really means. We're gonna see an example of that in a minute. The only spraying that I can think of is that you can stimulate flowering by spraying, or you can probably spray gibberellic acid to make the grapes bigger, that we'll see in a minute. And you could, but abscisic acid has to do with abscissal formation. It is pretty much very specific. And of course, ethylene gas it's the only hormone that actually truthfully is a gas when it's applied. These others are liquids. So now with this slide, at least auxins, gibberellic acid, and cytokinins, these three, you'll notice that there's a benzene ring in all of them. There's a pyrrole house, I call it, upside down house here and sideways house. That's basically a 5-sided polygon that has a nitrogen in one part of it, at least one nitrogen. So a pyrrole, Pyrrole molecule, and a benzene ring are fundamental to some of these structures. But that's why indole acetic acid looks a lot like one of the 20 biologically important amino acids, tryptophan, okay, which is the root molecule is similar in both cases. Now, before we leave this slide, I want to say that there are synthetic growth regulators that technically aren't considered hormones. These are manufactured hormone, and hormones can be termed growth regulators. We've already said that, but synthetic growth regulators aren't labeled as hormones. They're just called synthetic regulators. 2,4-D is a good example of that, and so is 2,4,5-T, which has to do with similar, if I'm not mistaken, to Agent Orange. But now those can cause abscission or defoliation. But so they're synthetic auxin herbicides that developed and used for weed control. And the reason that here's something that's interesting. Auxins in large amounts, are toxic to plants. And they're most toxic to dicots and less so to monocots. And because of this property, synthetic auxin herbicides can be used in weed control for if you've got corn as a monocot. So you can cut out a lot of dicot weeds. I'm not saying spraying herbicides is a good thing. I'm just saying that's the way the physiology works. And that's why it works that way. Now, we're going to go into plant responses, plant movements, tropism, and nastic movements. Plants, this is away from the hormones more in terms of responses. I've actually already told you a good bit of it in saying that a tropism, trop is Greek for change, and there irreversible changes that take place. We'll talk soon about phototropism, gravitropism, and thigmotropism, but those are the three major types. Well, what happens in a tropism is the cells on one side of the plant elongate more than the cells on the other side, causing the plant part to bend towards the source. The side, it'll bend toward the side with less growth. Let's go ahead and go to these here primary. Phototropism is allows the plant to maximize light exposure, and that's what's going on here. Here's the apical meristem bending toward a candle, but so maximizing exposure to light, which plants require for photosynthesis, and it really minimizes it in plants subjected to intense light and heat. Gravitropism, so I've got okay, this is like an example of gravitropism, allows the roots of a plant to determine the direction of gravity and flows downwards. And involves, what we're seeing here is something called statoliths. Statoliths, and this particular example of a statolith is starch grains. And the starch grains have migrated from one part of the cell to another part, and this is functional in the bending mechanism that goes on in geotropism. Now, the third, thigmotropism is, let's see, this is not the slide I wanted. Let's see if I can find. All right, okay. Thigmotropism has to do with touch. And so when certain vines touch object, they respond accordingly. Some vines go clockwise and some go counterclockwise. We're talking about the fact that the two exotic wisterias, the Asian and the Japanese, one, I don't remember which, Weekly I think has it, one of them bends clockwise and the other bends counterclockwise, and that's useful to help you to determine which is which of those two non-desirable wisteria vines. I don't know which way I forgot which way we saw it this weekend, past weekend, which way the native azalea, native wisteria grows. Now back to, where was it? Back to here. Let's see if I've covered everything. All right, well, we're going to talk more about nastic movements in a minute. I already told you about the three primary differences. The one nastic movement I haven't mentioned, though, is the way the Venus flytrap works, and that's a nastic response, a turgor water response. Now, all right, this is which is a thigmotropism, which is a thigmonastic response. I've kind of already spilled those beans. These are both legumes, and this is the This is actually a thigmotropism, like very much like what's going on the right-hand side of this slide here. The same response. This is a galactea or milk pea and responding to a thigmotropism type of pattern of growth. Looks like it's going clockwise. But there is tiny little structure here called a pulvinus that you can't see over here on this other legume. And that pulvinus on this plant on the left is involved in a turgor movement, a movement of water. So this kind of like a touch me not, well, not the same, but when some of you have seen the sensitive briar and other legumes can do it, where you touch the leaf, in this case it's a doubly compound leaf, the whole thing here, and the finger in this case unfortunately is covering up the pulvinus, the pulvinus, and that's where turgor movements or water movements cause water leaves the leaflets and they droop, and it may take 2 to 10 minutes for the water to come back depending on the availability of the water. But as we already said, a nastic movement, they will return to their former condition. It may not be as quickly, but they will return to the former condition, unlike a growth movement. Once this growth takes place, you could peel this vine away from the stem and the pattern would still be there. The growth is irreversible. Now, sleep movements, NYCT. NYCT, oh, by the way, last thing is, as we said before, thigmonastic movements are independent of the direction of your stimulus. You could the finger comes from the other direction and you get the same response. Sorry about that. Now, nastic movements, as we just said, are non-directional to the stimulus. The temperature, humidity, and the light, it doesn't really matter. But if you cut the light down, nicto means night. And we're referring to, like in this wood sorrel, a sleep movement, when the light drops down, the leaves start to droop in certain plants. And that's what's going on here. There is a family called the Nyctaginaceae, Nyctaginaceae, and that's the scientific name for the 4 o'clock family. And the 4 o'clock family is known for having opening its flowers at certain times of the day, usually in low light situations. But there's photonasties, that's a response to light, not dark, like nicto, they're responding to dark. Sleep movements, that's another name for nyctonasty. There's chemonasties, responding to chemical or nutrients. Hydronasties, HYDO, responding to water. Thermonasties, responding to temperature. Geo/gravinasties, responding to gravity. And we've already talked about the thigmonasty, responding to touch, like you see on the left here. Let's see. All right, this is a member of the bean family, bean, actually showing a thigmonasty, and saying a nyctonasty, excuse me, I say thigma, I meant nyctonasty, is circadian rhythm movement of plants in response to the onset of darkness. And we saw it in the wood sorrel, now we see it going on here. Okay, saw it going sleep movement. This is a thigmonasty, or excuse me, a nyctonasty. All right, we already talked about this slide and the 3 major types of tropisms, the most commonly talked about, photo, grava, and geotropism, non-reversible movements. And this is an example of phototropism, geotropism, and this is actually, okay, this is showing what happens to apical dominance, like I described with my southern swamp, my swamp oak. When you cut the apical meristem out, there's no longer suppression of these lateral buds. These axillary or lateral buds have been suppressed by the terminal bud. And when you remove the terminal bud, the lateral buds are allowed to exercise their growth. They're being suppressed here. And this has to do with reaching up for light. So even though the plant doesn't have a brain, it's got a strategy to get to light, especially if you're in a dark forest floor. You don't need to get sprawling out everywhere. You need to go up. And that's what apical dominance has, the practical ramifications of apical dominance in the forest floor if you're in the understory seeking light. Fortunately, my swamp white oak is in not having to deal with the light problem. It's out in the open. Now, as I said back on the table slide when I was showing gibberellic acid, I told you need to add that one of the things that gibberellic acid can do is affect fruit development. The one on the left was a control, not sprayed with gibberellic acid. The one on the right was treated with gibberellic acid on these grapes. Plant response, this has to do with ethylene, which is the only hormone and plant hormone that we talked about that is applied as a gas. And there's 3 little situations here. Number 1 is with ethylene releasing orange, a banana was put in a bag. And then second one, banana was put in a bag with a liquid. Well, actually what happened is the liquid volatilized as a gas in the bag and ripened a banana, really over ripened it. And then the third one is the so-called control, where they just put it in a plastic with all three with equal time in a plastic bag. So time was not a variable. Now, what I would have done, in addition to that, instead of sticking this number 3 control in the bag, I would have had a banana separate, not in a bag. And I can assure you that a banana that's not in the bag is not going to be as dark as the one in the bag, because the reason this number 3 control, so-called control, as dark as it is the banana itself produced some ethylene. That's what, but it got speeded up by being confined to being in the bag. So ethylene's involved in all 3 of these because they're all 3 were in the bag. Questions? Now, a little deceiving here. This number 2 shows you a liquid ethylene, but what happens is it evaporates quickly into the air and causes the effect. Now, I think this relates to Rana's question, or well, this is what I was trying to get at, when the only application I know of in nature that refers to leaf abscission or to abscisic acid is this one. And abscisic acid concentrations is what forms a layer of corky tissue forming between the petiole on the right and the stem itself on the left. And as I said, if you artificially extend the photo period beyond what would normally happen at the end of the growing season, you can keep the leaves of the plant on longer. And that street light situation proves that in the temperate zone at least. Well, so we have 2 factors going on here. Not only the abscisic acid, the word that does not appear on this slide, but the fact that the auxin production is dropping. So it's really a combination of 2 plant hormones going on at the same time in combination with environmental cues. All right, breaking bud dormancy, abscisic acid is a part of that. At the end of winter, abscisic acid concentration drops and the buds start developing. Now, we saw, I brought up on a field trip this past weekend, one of the real reasons that people have trouble identifying woody plants is because they can't tell leaf arrangement. And the best example that I showed them was a plant called gum bumelia or Sideroxylon lanuginosum. Bully is a common name, and it has spur shoots. Pears have spur shoots, apples have spur shoots, ginkgo has spur shoots. They're basically shoots or stems that are accordionized versions of a stem that if you elongate them, you'll get a normal stem. And so because of that kind of spur shoot development, it's hard to tell the leaf arrangement. You have to find the new growth to be able to do it very effectively, because otherwise they look whorled when they're WHORLD when they're really not. And so what's happening is we got an accordion on the left, then the accordion is stretching out at B, and the abscisic acid plays a part in that, and the concentration drops, the bud starts developing. Now, with that, I'm going to put off the last 4 or 5 slides till next time, and leaving us a little time for dessert, which today to me is that something I've been promising we would talk about, the Polyclave key concept. Are there any questions before I drop out of this PowerPoint? All right, hearing none, I'm gonna go ahead and bring up the, I'm gonna actually close out this PowerPoint, bring up the Excel, the VPT, Cancer Vascular Plant Taxonomy. And I'm also got down here, I thought I had, here we are. These are the 5 pictures that I sent you. Okay, this is number 1. I guess I'm gonna call it number 1. The second, actually, this is the first one. It shows something that's really not enough. This is where I screwed up, and I'll tell you how that is. All right, this shows other features. And actually that that right there, where those 5 stamens are pointed toward one another, almost triangular and covering, if you looked at it from side view, it would look like the oleander model that I showed you way back when we talked about reproductive structures. But I shouldn't have said that because it gave you a hint about what family this is. This shows that what we're really doing is plant analysis. And plant analysis with a flower structure is a little more involved than plant analysis for a tree or shrub, because you're looking at vegetative features and reproductive features. You can tell from this slide, though, that these petals are not love me not types. They're not polypetalous. They're gamo or sympedalous. The petals are fused. So we have corolla tubes that have lobes they're not separate. They're not polypetalous. And that's important when you try to identify plants. And this one doesn't show a whole lot except for something that's typical of the family, and the way that cone looks when it's closed. So that doesn't. Now I got these from CERNAC, which is the Southeastern Regional Consortium. They have a plant identification thing going on every day. I believe every day they switch it. And I already pulled out another one that I won't do this week. But what I would ideally like to do, if this was going to work, we'd all be in the same room and I would all handed you an unknown plant, or at least one you didn't know what was. And I would, this Polyclave key concept I've talked about, it works like dichotomous by eliminating, but it works, you can enter it at any number of places. Now, I sent this to some people, and if you want it and you don't have it, I'll send it to you. And in the notes section down here, I'd like you to read it before you do anything. One of the things is saving it to a hard drive or desktop would be good on a CD, since you can't change it or mess it up. The bottom line is, I would have, if I were you, a spare of this in case you ever screwed it up. I haven't locked any cells, so it's easy to mess it up. Navigation is easier with a keyboard, arrows, and a mouse. That's a matter of opinion, but I think so. When done, don't save anything, close it, and return to show all after filtering. All right, now these others are notes that I made. There's column P is red until tested, and that's column P is the newest one, and it's still undergoing testing. We're going to use column P today, and you'll see why it's still under testing. All right, so to identify, first of all, this is not a key to species. It's not a key to all families of plants in Georgia. It's a key to the dicots in Georgia, dicot families in Georgia, of which there are about 200, let's get down. No, there's not, 148. There's 148, which in an Excel file, the top row doesn't, it's not being counted here. So there's 148 dicot families in Georgia, give or take, depending on whether you include certain exotics or not. But I've excluded certain families not in Georgia, and these are mostly the ones that are found in Florida, here by Florida, and some of them are just others I've used in my lectures from time to time, like the largest family of the flower in the world is Rafflesiaceae. So I don't know why I bothered putting it in here, but anyhow, to start this process, you normally would hit Control Home, and for a Dell key, your keyboard, which is, I call it this thing, I've got the Dell from hell. You have to hit pretty well to make it happen. Control, Function, and Home to make it get the word to the top. And this, if you look at these left 2 columns, I froze this sheet so that they'll stay there. That's on purpose. First column really is not important to you folks. The second column is the one that's important to you for this purpose, because in a Polyclave key, one of the nice things is that you say, I don't like stipules worth a flip. Well, you can skip them. You can not pay attention to stipules. If I say, well, I can't tell superior from inferior ovary. Well, you can skip that for a while, but you eventually will have to come back to it. We won't get to that because the second part of this table involves reproductive structures. The first part of the table involves vegetative structures. And that's how we're going to start. Any questions before I start? I went to control home. And one of the things I want to do is determine if the plant is woody or herbaceous, C or D. And by the way, a friend of mine looked at this and said, this is okay, but you could do a lot better if you put formulas in it. And I'm not that good at math to do it. I can do a few formulas, but his brother is supposed to be good at it. And I'm talking, John, I'm talking about Mike's brother, Mike Owen's brother. Anyway, Mike liked it. I showed it to him, but he said you could do formulas. And, you know, you could do a lot more with this, I'm sure, but it serves my purposes pretty well. All right, control home. I'm at the beginning location. And I'm going to give you a hint here. This is where I actually screwed this up because I thought when I saw this picture that this was a woody plant. Now, it turns out these are rocks, and these are lichens on them, and this is not a woody plant. It's a herbaceous plant. So if you actually had the plant in your hand, which was what we would normally do, it would be easier to make that determination. There are other things that you could determine too. We'll point out at the very end that if you had the plant in your hand, like crushing the foliage or if there's milky juice or not, you could determine that you can't determine by these pictures that the Cernak has provided. Okay. All right. So I'm going to give you the benefit of what my mistake was, and I'm going to say it's not woody. I'm going to come over here. This is set up in filter mode, and I'm going to select on this, and I'm going to do I could do text filters, which is almost foolproof because that takes care of exceptions if you do it right. But I'm going to go down here and click select all, so I deselect and just select the X. Now, when you see an X in parentheses, it means when it's the exception. And so, and that means by selecting the X, I'm leaving these other two categories out. And so you say, gee, why don't you just go up and do text filters and say contains X? That would be the one way of doing it. But I'm going to cheat and just select X. Now, what I want you to do, I told you there were how many families? 148. And I'm going to, after clicking OK, notice that we're only dealing now with how many? Bottom left says 87 of the 148. So I eliminated a good many, 50, 60 different families. Now there's no special habit involved here. It's not aquatic. The next thing we look at is leaf arrangement. Now, ideally, if you're keying by yourself, you would have done plant analysis to the point that you wouldn't have to keep looking. This looks opposite. Let's look at another slide to see if we can determine that. I thought I saw one. I know there's a better one, a better view than that. Here it is. It's a little out of focus, but this clearly shows, along with the next slide, that the phyllotax, your leaf arrangement, is not alternate, not whorl, but opposite. Now, with that in mind, we can go over here and say, all right, we're dealing with 87 on 148. Let's go select all and select X. And then let's go here. And we're down to 34 of the 148. So keying, including polyclave keying, is about eliminating, process of eliminating. And what you're left with is provisionally the plant your unknown is in these families in this list of 34. Now, based on the photographs that we've been given by Cernak, we can't, or at least I can't, tell about the stipule situation. And I was wrong. We will be doing some floral stuff, and we're going to do it now. Perianth is present, so you skip that. It's not apetalous, which means another when there could be petals and yet there's still sepals. And actually, I've already told you, spoiled it a little bit by telling you it's not polypetalous, it's gamma or sympedalous. Let's go back and review that. Anybody got questions so far?
Yes.
Okay. Steve, yeah.
Would it help to further eliminate other possibilities by going to like the opposite versus alternate? Go into the alternate column and select blank.
Well, you wouldn't go to the alternate column, Steve, because these are opposite, and we've done that.
Right, but you see all the records that are showing that have alternate?
Yes, yes. But all right. I wonder if it would have been, yeah, I see what you're saying. But what these are plants that can have both.
Ah, okay. So you wouldn't want
And
to
if we had
by
gone,
eliminating some.
you know what, we could back up. That was the last thing I did. Okay, we've got 34 showing. Let's go back. Okay, oh, it's not really going back. Well, says it's still showing 34 and it shouldn't be. Instead of doing, I'm going to try something different. I'm going to go, instead of going select all, I'm going to go text filters and I'm going to do contains X and see if we get anything different. Let's just see if we get different. Still, okay, instead of having 34, we got 43, which is not as good. But all right, we for some reason that did that. I guess that brought in some more that the other way. Let's see, this way I didn't take care of that's right. I'm not getting rid of the exceptions this way. I thought I was. But anyway, we're going to go into, all right, this is not a polypetalous, it's gammopetalous. And we're going to, this time I'm going to select this and drop down to 19, okay? We've got 19 of 148. Now, we could use this stamens. Actually, we could do flowers regular, but I don't think it's going to give us but maybe one advantage. Let's see. We got 19. It might drop down one. No, 10. Okay, it dropped down quite a bit. The flowers have regular symmetry. Okay, so that helped us to narrow down. They weren't like a P bilateral. You know, if you cut this that way, or that way, that's what you call actinomorphic or radial symmetry. They did get us down to 10. I didn't do that when I was practicing, but what I'm going to do now, what you could do at this point, and I wouldn't recommend it, is you could go over here and look at the semi-unique character states, or most importantly, the keywords of diagnostic information. That's the nutshell information about every family that's on here. I'm going to back up though. Let's not do that. Let's try to get fewer than 10. Let's try to eliminate some more. And we're going to go into the red zone, which is normally it's red because it's not been tested by students very much. Most of this dicot key, unlike my monocot key, has been tested by students. And I've worked out most of the bugs. All right, I'm going to go into here and I'm going to go through text filters, and I'm not going to hit equals. You think, well, a number, why don't you do equals? Well, I've learned that contains is a safer bet. I'm going to hit contains and custom, and I'm going to go for what we just counted. You saw 5 stamens, right? Okay, so I'm going to select 5 and then see what happens. Okay, now we're down to 4 out of 148 families. Now I'll go back and, okay, see that there's 5. 1, 2, 3, 4, 5, no matter which way you go, 5 stamens. All right, just reaffirming that in case you missed that. All right, so we got 4 families. Now that's a typically good situation to be in. Most is in, well, you can't really respond. Well, you can, but the Valerianaceae family is and let's see, I thought I had a place that actually told how many it was in Georgia. Maybe not. Let me look in here and see if I've got a number. Bottom line is there's not many in that family, and I've got one in my yard. It's a weed. I thought I had the number in Georgia there, but I didn't. Let's see, keep going. Okay, here we go. Let's see. No. Whoops. Yeah, here it goes. In Georgia, there's only 3 species. All right, opposite leaves, somewhat juicy herbs with small white epigynous flowers. That means inferior ovary, and quite like inflorescence, valerianella. It's a salad, some kind of salad is a common name. But anyway, that's not our plant, and they're not strictly white. It doesn't fit. They're not small, juicy herb. Well, they're not small flowers. You really can't appreciate what the flower's like. All right, now let's see, the gentians. Okay, now gentians, most of them flower in the summer or fall. And this plant, you'd have to know, is flowering in the spring. So that, and it's, let's see, herbs, leaves opposite, estipulate. We don't know about the stipules on our plant. Corolla tubular, that's true. But the cavities aren't right in our plant. And we don't know, we can't see the way the seeds are inserted. That's what's called placentation. But, and there's 26 species of gentians in Georgia. But using HipDad for P for phenology or flowering time, and I'm telling you in the spring, and there are some gentians that flower in the family that flower in the spring, but their flowers are almost microscopic. So, and they're not very common either. Most of you've never seen them. So that's part of HipDad too, abundance. They're rare. Now, this family, my favorite family really, the gentian, the Rubiaceae is easy to tell because they have interpalleolar stipules, and the family we deal in, this unknown is, rarely has stipules at all. The problem is our plant, you can't see the ovary. Well, maybe you can. All right. Bottom line, our plant does not have an inferior ovary, and the leaves can be opposite or whorl, like button bush can be whorl. But let's the ovary is under here. Let's see if we can see something. It's so much better if we had the actual plant in our hand. There is one that gives us a hint about the ovary position. I'm trying to show you our plant. All right, you can't see the ovary, but this is where the ovary is, and you'd almost have to guess and say this is a superior ovary because it doesn't have a hypanthium. A hypanthium is when you have a few sepals, petals, and stamens all together and form a tube. You know what? Well, I'm not gonna, my desktop, if I could get to it. Well, I have I call it Donna's hypanthium because I was explaining this to Donna one time, and I have a slide that maybe if I remember to show you before I go, I'll show you what hypanthium look like. But bottom line is the key. All right, here, let's go back to the keyword semi-remarks. All right, semi-unique character states. All right, broken tissue with milky latex. If we could take this plant and break it, it would be copious milk coming out of it. And that's the only one that's got this. And so this is in the dogbane family. This is Apocanum andricifolium, which is, you think of the common name, but that's the plant. Apocyanum or Apocyanum, Apocanum andricifolium. And this is a northern Georgia plant. That's what the plant is. And the ovary, if you can't see it, is half inferior. But all right, stipules you're mostly, you wouldn't see them because, and when you do, they're really inconspicuous. So that's how that works. Now, the best way to do this, I'm going to go out of here and don't save it. And I'm going to go out of this and see if I can bring up my desktop while I'm at it. Yeah. Donna's hypanthium. Well, that's not the, okay, let's see. That's one of them. Let's see if I've got the other one here. No, that's not the one I want. Let me get, all right, this is the one I really wanted to show. This is a prunus. It has a hypanthium. What you've got here is you got the fusion. This is how you go about telling superior from inferior ovary. This is a superior ovary that's got a moat around it, basically a section. You got a fused calyx, corolla, and stamen. The stamens are fused into one structure called hypanthium. And whereas in our plant, they were separate. So that's one way you can tell a hypanthium when it's present. That's a perigynous situation. This one is this is like an apple where you have the hypanthium, but it's fused to the ovary wall. There is no moat around it like in the one I showed you just now. They both have one thing in common. They have hypanthiums, but this is an inferior ovary, and the one I showed you with Donna's qualifies as still a superior ovary. All right, let me bring back up the, get rid of the animation tools. Stop sharing the screen. And are there any questions? Well, we went over about 15 minutes, but that's why I'm glad we stopped doing what we did when we did. And I think I'm still, it's funny how y'all can tell. I can't even tell by, I guess I could. I'd like to be able to see a number of people that are there, and I know everybody's here, but Donna was able to see it. Okay, I see it, 8 at the bottom. Okay. Anyway, I didn't hear much out of anybody, so I hope I didn't lose too much of anybody. And if you got any, I guess I could look in the chat and see there's nothing there. That's probably a good thing. All right, next time we'll deal with the remaining few plant physiology slides, and I'll go into my favorite mode, plant reproduction. And again, it's not about sex. It's about the idea that people think it's hard when it's really not. And I'm not sure we'll finish all of it, but we'll start it. And we'll compare the plant cycle to your cycle and one other cycle, the kind in that. And there's 3 fundamental sexual cycles in all organisms on the face of the earth. And everyone can be pigeonholed into that. Okay. All right. Well, thank you very much. Not much dialogue here. I got Donna with a thumbs up or no middle finger. Okay, good. All right. So, all right. Have a good rest of the weekend. Steve got a thumbs up and Jackie did too. All right. Bye, folks.
Thanks, Bobby.
All right. You're welcome.
Thanks.
Hey. All right. I'm going to.
Bye.
Got something, Donna, for a shutout?
Night.